change the size.
So, I'm going to add a few more words about electron transfer and then move on to proton. So let's see, we talked about these 3 of the 4 complexes, and I'm pointing out that they have these prosthetic groups that are tightly bound to them. And I went into a little bit of detail by complex 1, which is the only one I'm going to go into detail on. And I did point out that there's a sequence of electron carriers. You don't have to memorize, although it's not really that bad. It's only the flavin and then the iron sulfur clusters to get the electrons from NADH to coins on Q. I just thought I would mention that because then in complex 3, which I did not go into much detail on, but I did want to just point out that in complex 3, electrons have to go from reduced coins on Q to side group C. And there's, again, a series of electron carriers within complex 3 that does that. And those electron carriers are those prosthetic groups that are listed in the table. And so it's a similar idea of electrons being conducted through the protein by those carriers. And the same goes for complex 4. There's some electron carriers in complex 4. And so they're listed on the table. I think there are hemes and copper ions. So, as I said, I don't plan to say anything more about complexes 3 and 4 beyond what I did last time. All right, now, here's what I do want to just point out, is that these are all redox reactions, getting the electrons from the NADH to the oxygen. And so I just thought I would point out that the components of the electron transport chain are listed in this table of reduction potentials from chapter 13. And so, for example, complex 1, the overall reaction is NADH plus coenzyme Q. So that's the overall reaction. So we'll find the numbers that we need in this table to allow us to calculate the delta E for this reaction. So the delta E 0 prime is going to be E 0 prime for one of the components, and that component is going to be coenzyme Q or ubiquinone, which is plus 0.5. And we'll subtract from that the reduction potential of NAD plus, because NAD plus is the one that we're going to turn around. So E0 prime for coenzyme Q is plus 0.045. And for NAD plus, E0 prime is minus 0.32. So if we plug those into here, the delta E is 0.045 minus a minus 0.32, and that comes out to be plus 0.365 volts. And so the overall standard state free energy change of this reaction is minus n times F times delta E0 prime, and that's minus 2 times 96.48 times 0.365, and that's a large negative number. I think it comes out to minus 70.
Is it always -2 for n?
The value of n depends on the reaction, so you have to look carefully at the stoichiometry. So for these reactions, it's always going to be 2. Okay, so I just did that calculation for this reaction for using NADH as a reducing agent to reduce coenzyme Q or ubiquinone. So that's the reaction of complex 1. And so electrons are going from a substance with a large negative reduction potential, in other words, weak oxidizing agent, relatively strong reducing agent. They're going from a strong reducing agent to a stronger oxidizing agent, weak reducing agent, right? So that's what you need. You need to go up and to the left in this table to have a spontaneous reaction, to have a positive delta E and therefore a negative delta G. And so that's consistent with what we would expect. So coenzyme Q becomes reduced to ubiquinol, which is QH2. Ubiquinol has a slightly positive reduction potential, so it's not all that strong as a reducing agent. But the next one in the table is up here, and the transport process is here, cytochrome C. Cytochrome C has a more positive reduction potential. So again, reduced coenzyme Q will spontaneously reduce cytochrome C. Goes up and to the left. So cytochrome C becomes reduced, the Fa2 plus form. And cytochrome C, although it's not a very strong reducing agent, is strong enough to spontaneously reduce oxygen, again, up and to the left. So you have this series of electron transfers from initially a strong reducing agent to ever stronger oxidizing agents, ending up with oxygen. So that's, again, what one might expect for an electron transport chain. So here's another figure that I used to make the same point. I took this figure from another textbook, and then I corrected the reduction potential. So there's the reduction potentials again. So we're going from negative, slightly positive, more positive, highly positive. And I did the calculations for you. Again, there's the delta E for that step, delta G, and for the rest of it. So you can confirm at home that my calculations are correct. Note that each of these has a rather large negative. So this is the reaction catalyzed by complex 1. There's the delta G that I just calculated, large negative number, complex 3, and complex 4. So note that each of these is a large negative number. So free energy becomes available to do work. And the work, again, is the pumping of protons from one side of the membrane to the other. Okay. So the question that you're all asking, and actually someone asked last time, is what about complex 2? So I skipped complex 2, so what happened to complex 2? And here's another question, which I pointed out a long time ago, which is what happened to all those 2 electrons from the citric acid cycle, chapter 16, which were left in FADH2, right? So let's remember those 2 electrons. There they are. That's chapter 16. So in chapter 16, in the citric acid cycle, we learn that succinate becomes oxidized to fumarate, and the oxidizing agent FAD, which picks up the 2 electrons and the 2 protons from succinate and becomes reduced to FADH2. And we usually say that FADH2 is a product of the citric acid cycle, but as I think I pointed out, remember this is a succinate dehydrogenase reaction. Yes?
Didn't you say that the 2 electrons from FADH go to NADH?
No, I did not say that. Not for this example. I said that for pyruvate dehydrogenase and alpha-ketoglutarate dehydrogenase. I did point out that this FAD is a prosthetic group that remains tightly bound to the enzyme. So those electrons have to go somewhere. And the question is, where do they go? And the answer is that there's a series of electron transfer reactions, the intermediaries of which I don't remember, but ultimately they are transferred to coenzyme Q. And so the product of succinate dehydrogenase is not FADH2. FADH2 is an enzyme-bound intermediate. The actual product is reduced coenzyme Q. Okay. And this reaction, so in this figure, some of you may have noticed, I skipped over complex 2, but here it is, and there's succinate. So succinate is converted to fumarate. There's the flavin, the FAD, right? But what's indicated in this figure is that the electrons go from succinate to the FAD, but then they continue through various electron carriers to coenzyme Q, and reduced coenzyme Q is the actual product. Right? So that's the reaction catalyzed by complex 2, but complex 2 and succinate dehydrogenase are the same thing. Right? And so in chapter 16, we call it succinate dehydrogenase. In this chapter, you can call it complex 2, or there's another name in that table. That'll come up in a minute. And I don't know how this happened historically. It may be that people were studying citric acid cycle and others were studying electron transport. They gave them different names, and maybe later they could kind of do the same. But I mean, that's the story. So these are the same thing, but again, what's the overall reaction now catalyzed by this enzyme? The overall reaction is succinate is a substrate, and oxidized coenzyme Q is the other substrate for this enzyme, as indicated in this picture. And the products are fumarate plus reduced coenzyme Q. Again, that's a product, so there's the coenzyme Q. It's released from the enzyme into the membrane, where it can diffuse around in the membrane and eventually can find its way to complex 3. So it doesn't matter how the QH2 is formed, complex 3 is the one that's going to oxidize it back to coenzyme Q. Okay. So yes.
Does this succinate to fumarate complex 2 happen here, or is it
That's the reaction. I mean, so this is succinate dehydrogenase. That's the active site. So it's the same reaction we learned in chapter 16, except now we finish the reaction. So we get it. So here's the details, and again, the details are important, but this again sort of shows you it's another story. So here's the flavin. Succinate is going to bind. Electrons are transferred to the flavin, and then again some iron sulfur clusters until they reach coenzyme Q. And there's this heme in there, which is not directly involved, but it's the same story of a chain of electron carriers to get the electrons from the succinate to the coenzyme Q. So it's similarly as in complex 1. Okay. So back to this, yes.
So would you consider complex 2 to more be a part of the citric acid cycle rather than oxidative?
I think the answer is that the two are very tightly connected, and so one isn't really going to happen. Once those electrons are taken from succinate, they're going to be given a coenzyme Q. So they're not having an. All right, so back to this table. So here's succinate in this table, and so here's the reaction catalyzed by coenzyme Q. Electrons going from succinate to, reaction catalyzed by complex 2. Electrons going from succinate to coenzyme Q. And note that their reaction potentials are very close. So the delta E of this reaction is actually very small. And the standard state for energy change is also very small, slightly negative, but it's not a very big number. Who knows what it is in the cell conditions, but presumably it's also small. So, if I can add, I'll just add this here. I think this is the complete picture from this other book. So here's another name. You can call it complex 2 or succinate dehydrogenase, or you can call it succinate Q reductase if you want to. Those are 3 names for the same thing. But again, there's the thermodynamics of this reaction. So the delta G standard state is fairly small, perhaps slightly negative, but essentially no work is done. And the work that we're talking about is pumping protons. So in other words, no protons are pumped. So this oxidation of succinate and reduction of coenzyme Q is not accomplishing proton pumping, unlike the other. Actually, all 3 of the other complexes, remember, keep pumping protons. Okay. All right. So that's all I have to say about electron transport. Are there any questions before we move on to proton pumping? Yes.
For the diagram for complex 2, I believe it showed coenzyme Q exiting the membrane and then re-entering?
I think that was because in their drawing, complex 3 was in the way. So I think we'd have to go around complex 3.
So it's still going to be in the membrane?
Yeah, it's in the membrane. It's there's no way it's going to be in the aqueous. So I think that was just artistic inaccuracy. Any other questions? All right, let's see, I want to now move on to proton pumping. All right, so there's our old diagram. So before I do that, let's just, I just want to sort of count up protons. And let's consider an NADH, where the NADH, the electrons in the NADH eventually end up being transferred to half a mole of oxygen. And let's count protons. So in the figures, in this next slide, yeah, there it is. So complex 1 pumps 4 protons. Complex 3 is 4 protons per NADH. Complex 4 is only 2 protons per NADH. Yeah, they show 2 per NADH. So it's 4 per oxygen, but only 2 for half a mole of oxygen. Right, so that's 10 protons. In making these tables, we usually say, what about FADH2? So in this context, what we're really saying is for succinate, where we get this FADH2 as an intermediate. Actually, there's going to be at least one other metabolic oxidation pathway where we're going to get a FADH2, and it's going to be a similar thing. So the FADH2 has to be reoxidized by complex 2, which pumps 0 protons. But then the coenzyme Q that's reduced, that's formed by complex 2, is reoxidized by complex 3, just like any other QH2. And so that's going to be, again, 4 protons. And then again, complex 4 is going to do 2 protons. And so you get 6 protons pumped per FADH2, if you want to think of it that way. But again, it really means per succinate. So you don't get quite as many protons for that succinate as you did for the NADHs. That'll come back. We'll do some more accounting later. All right, so how are these protons pumped? So complex 1, complex 3, and complex 4 all manage to get protons across the membrane. Complex 3, I already said what happens for complex 3. Let's look at proton pumping mechanisms. So here's the membrane. Here's some oxidized coenzyme Q in the membrane. The coenzyme Q becomes reduced to coenzyme QH2, and as I said last time, those are protons from the matrix side of the mitochondria or from the cytoplasm of a bacterial cell. And so this is accomplished by complex 1 and also complex 2. Right, both of those produce QH2, and in both cases the protons came from the matrix or the cytoplasm. Either, regardless of which of these complexes made the QH2, it becomes reoxidized by complex 3. And as I said last time, the active site of complex 3 must be situated and situated. The protons that are taken off the QH2 end up in the intramembrane space of the mitochondria or in the periplasm of the E. coli bacteria. So that's the first pumping mechanism. It's a very simple mechanism. I said it. And the second mechanism, which is the one I want to focus on, is thought to apply for complex 1 and perhaps also complex 4. And so mechanism number 2 is a conformation change mechanism. You can say it's an allostery mechanism in a sense. It's events happening at one part of a protein which induce a change in another part of the protein. And I'm going to illustrate this using complex 1 in some structures that were determined recently for complex 1. I'm going to show some structures that came from, I think really this article, it's now several years old, but this article they actually proposed the mechanism. They determined the structure of part of complex 1, and based on that mechanism, based on that structure, they proposed the mechanism. So I think before I get to that, I want to just talk a little bit more generally. So before we had that mechanism that I'll show you in a minute, we had to make just very vague cartoons. So let me give you the sort of vague cartoon picture first. So here's the membrane. And let's see, I think in my diagrams, this is the matrix side. So this is the N side. Remember, that's the side that's going to have a negative charge. And so this is the P side. This is going to have a net positive charge because protons are going to be pumped from the N side to the P side. So there's protons up here on that side, but there's more protons down here. So put a bunch of H plus in here. And so we're assuming it's an ongoing metabolism. All right, so a cartoon version of a transporter might be something like this. That's a protein. It's embedded in the membrane, and it can be open to one side. And there's some group on this protein which can bind to protons. So there has to be a base in this enzyme. And so the base is actually thought to be a lysine, one or more lysines. So there's a lysine side chain. So it can be a base, but lysine, of course, can grab a proton. So if it grabs a proton, it's protonated. Of course, that protonation reaction, just like any other protonation reaction, is reversible. So it can grab protons, let go of protons. The conformation change idea is that this protein can undergo a conformation change such that whereas up there it's open to the matrix side, it can somehow undergo a conformation change so that it now is open to the other side. And so the lysine that was sticking out into the matrix is now sticking out into the intramembrane space on the P side of the membrane. Again, there are protons on this side still, but there are more protons on this side. That's the P side, assuming that there's sort of ongoing metabolism, protons are being pumped. Right, this conformation change is reversible. This is it's a protonated lysine. It's an acid. So what do acids do? They give up their protons. So this has a chance of giving up its proton. There's already a lot of protons here, but there's no reason it can't give up its proton. This reaction giving up the proton is reversible. So we could just as easily do that. It may be that the protein with the unprotonated lysine can also undergo the conformation change, right? So that lysine can switch back and forth across the membrane. So those are the steps. So we want to use this. This clearly shows a way to get protons from one side of the membrane to the other. But the key thing is we want to use this as a pump, right? So a pump means something where you use energy to move something where it doesn't want to go. Like you're pumping water, you're fighting against gravity, right? Here you're fighting against a concentration gradient. All right, so here's the protons we're talking about. It's these 4 protons pumped by complex 1. And this is the cartoon version of how these are collected across the membrane. So before we get to the details, let me ask a clicker question. So if you have your clickers, I want us to think about what this diagram implies. All right, so here's a drawing of this thing that I wrote on the board. So you can see this is the matrix side, the N side, here's the other side, the P side, there's the lysine, and I didn't show all the steps. Here I've shown it with a proton, but binding and release of protons is reversible. Note that there's a bunch of protons up here, but there are more down here. In other words, the concentration of protons is greater on this side than on that side. All right, and then all I'm going to show in this picture is the conformation change. All right, so there's the acid dissociation reaction on the outside. There's the conformation change. Now the lysine faces the inside, and it can undergo the same acid dissociation reaction on the outside or the P side. Right? So each of these is an acid. Potentially is to give up their protons, or these combine protons. So the question is, if this is going to be an effective pump, which one of these has to have the lower pK? Okay, so you can have the lysine on the inside. It's an acid. It has some pK value. You can have the lysine on the outside. It's an acid with some pK value. So which one is going to have the lower pK? This is related to Chem 103, Chem 271, Biochem 461. So here's your possible answers. I thought about putting I don't care on here, but I decided that's not a valid answer. So
Oh wait, he said so.
Wait, because that could be also. We want it to drop off the protons on the inside. So we want it to be more acidic. When it's missing the outside. Yeah, that's my thought. So it depends on how you word the question.
We can try to do that.
Well, I was wondering
I'll tell you. Like in the morning, walk to talk to the miss. So I'm making things up in my head.
Oh, no,
Higher PK. Higher PK. The lower PK.
Okay, you can enter your answers.
Which must have lower
So, there's your possible answers. Same question. It's not good.
You stand right up and goes.
yeah.
Okay. We're in the matrix and you want to go to it has to rather than go here.
Yeah, it's not letting us join.
How many of you are using this password? Everybody gets all the points.
Points.
It was set up a second ago before something.
If you write your answer on a little slip of paper, and give it to me before I reveal the answer, I will put your name on it. We'll have to do that for the second one also.
Yeah, I mean, I didn't answer
The 30-second thing, a lot of answers are in already, so everyone knows.
What are we saying? Which must have to, which would be more acidic?
Lower
More acidic.
There's like KAs and equal to like right? So it's like,
Use a larger slip of paper than that.
It's called conservation.
I don't know, James, like what if the lysine,
What'd you put?
when it's spacing the size near one of the electrons here, so it's stabilized by negative charge, the electrons is going to grab them up, and then as soon as you switch, you lose that, and then it just falls off. So it's not as much that the outside is significantly more acidic, it's just being significantly more basic.
well, you also said earlier, yeah. But like you also say that like you have,
Yeah, I can't read other
so that can also be
Anyone else?
people that
Well, that's what they're using.
It's supposed to be one more in a minute, so get a. Is that everybody? All right. So we're gonna have to totally won't wait. Everybody's going to answer it. All right. So let's skip the 30 seconds. So everybody pick D.
Well,
So
yeah.
almost everybody got it. It does matter. What if the PKs were the same? So here's the thing. What you need is that on the matrix side, you can think of it as, I express it as a PK, which has to do with the strength of an acid, but of course it relates to the strength of a base. This is a base. And the one down here is also a base. And you want this base to be able to grab protons, but you don't want this base to grab protons. So this base has to grab protons when there aren't very many protons. This base, you don't want it to grab protons, but there are lots of protons. Right? So the reactions, well, if we write it in the way I phrased the question, this base had better be stronger than that base because you need to grab protons on the inside. And moreover, again, you need to grab protons when there aren't very many protons here and not grab them when there are lots of protons here. Right? So you want this to be a strong base, which means you want this to be a weak acid. Here you want this to be a strong acid, and therefore a weak base. How strong, how weak is another question, but this better be a strong acid. because it needs to give up a proton. It needs to give up a proton when there's already a lot of protons. So only a strong acid will do that, a weak base. So you want the base on that side to be, this better be weak. And this is all relative. This base has to be weaker than this base, and this acid has to be stronger than that acid. So a strong acid means low pKa. So as most of you realized, this one better be the stronger acid, therefore the lower pK. What if they were the same? What would happen in this reaction as I've drawn it if the pKa's of these groups were exactly the same on both sides? What would be the net effect of this?
It won't be able to fight against the concentration.
That's right. So what would it do? Rather than fighting against the concentration gradient, it would just go with the gradient. And all it would do is you would get net transport of protons from this side to that side until the concentrations were equal. All right. So for this thing to be a pump, it has to be able to, the lysine or something has to be able to go from one side of the membrane to the other. That's the conformation change. But in addition, it has to have this more, I would say, more interesting and subtle property, okay? That the pKa of this lysine has to be different in the two conformations. All right, so now my question is, and get out your second half of your sheet of paper. I'm going to show you two and only two lysines. And I want you to tell me which one of them would have the lower pKa. All right, so here's a lysine. And I'm not showing you the entire protein, but this is a lysine residue in a protein, and this is a glutamate residue in a protein. And so let's say that. And let's compare that to this, where here's, again, a lysine residue in a protein and a glutamate residue in that same protein. And so of the two, which one has the lower pKa? Okay, so there's only two possible answers here. So first of all, there's no half point for getting it wrong. Sorry. On the other hand, as you start in your answer, you'll see, and so come to a consensus. Right now,
Why is
we're not coming to a consensus.
Yeah.
Which is typical.
Why is it
But the question is, who's putting in those answers? Do you trust them? Right, and remember, what is lower pKa? It means stronger acid, right? So which of these lysines is a stronger acid?
Come to me.
Is that bigger and better?
Wait, why?
Let me qualify this and maybe perhaps clarify this.
Okay.
Here's the reaction we're talking about.
That's what I thought.
We're talking about the lysine acting as an acid in the way that we normally think of acids. And the way we normally think of acids, we don't typically, we're talking about transfer of the proton to water, right? That's implicit. Over there, I didn't write water, but we understand it. It's not H+, it's H3O+. Let me write the water explicitly. So it's the standard acid dissociation reaction. I guess here's why I said that. It's not like the proton is going to be transferred to the glutamate or anything like that. It's lysine, it has to lose the proton to water, and the proton goes away.
It's all over now.
I don't get particle credit.
Because it's further away.
Oh, you get your reputation, but you know how like we normally get it wrong. Wait, is it 1 point of
Very relaxed.
Uno más.
It's literally one.
It's good to resist peer pressure, but not always. Everybody got an answer?
risky, too risky.
They change their answer if they so wished.
Or picture.
What is
That auction is having an event.
That was exactly that.
So we said 2%.
That
The
one
answer
first.
is,
What if it wasn't?
I'm sure the answer is B. I apologize to those folks.
Top 2% finally.
I'll
I'll keep my file count.
This is one of those things where having the negative, the strength of this acid has to do with how stable is this acid compared to a conjugate base. Having the acid just sitting there, it has whatever its strength is. If you put a negative charge very close to that, having a negative charge right next to the positive is going to stabilize that positive charge. And so that's true in hemoglobin, which is the only other example that is ever covered that I'm aware of, but it's also true here. This negative charge would stabilize the protein, and that would make this a weaker acid. It would make it hold on to the proton more tightly. Another way you could think about it is it's almost, you know, is that you've got this negative charge, and typically they're going to be hydrogen bonded to each other. So the glutamate may well be helping hold on to the proton. So both are now grabbing that proton and holding it there. And so the answer, I believe, in this case would be. All right. So all that is introduction to now how complex one works. And I don't really have time to go through all of it, but I'll try to. I'll at least set it up. And it won't take up long. Here's the structure of complex one from E. coli. So here's the membrane. Here's the part that sticks out in E. coli, sticks into the cytoplasm. If this were a eukaryotic protein, it would stick into the matrix of the mitochondria. Eukaryotic proteins have very similar structure. So presumably this mechanism applies generally. So there's a part that sticks into the cytoplasm or into the matrix, and there's a part that's embedded in the membrane. That's shown on the figures from the book. Here's the matrix side, which is the N side. Here's the P side. So protons have to go from the upper to the lower side. There's the flavin. So let's see, I've labeled some of this. Yeah, so there's the flavin that accepts the electrons from NADH. You can see the iron sulfur clusters, the orange things. And down here is where coenzyme Q is bound. That's where it's reduced to QH2. Right, so that's the reaction that I described last time. Electrons go from here, they sort of go from one cluster to the next, and eventually they get to coenzyme Q. Now, all right, so here's a cartoon version that now they've made this to show the details of the mechanism. So it's oriented in the same way. Oh, okay. So let me describe these membrane bound proteins. So the membrane part here, these are color-coded, but they're different protein subunits. So there's a sort of salmon colored, green one, a blue, yellow, blue, purple. Right? So these three, this purple one, the blue one, and the yellow one, each are going to provide a channel through which protons can flow. The fourth proton is thought to come through somewhere over here. I think the detail's not as clear. The other thing I want to point out is that this purple subunit has an alpha helix that extends all the way from this end all the way over to here. All right, so electrons are going to go down here to coenzyme Q. What's going to happen is that as the electrons are passing through the protein, there's going to be a conformation change that starts here. And that conformation change is going to be propagated into the membrane-bound subunits. And it's thought that this long purple alpha helix is going to be part of that propagation. So the movement of the protein here is going to pull on this alpha helix, and that's going to cause changes in the yellow protein and the blue protein and the purple protein. Right? So that long alpha helix is, that's going to have to move laterally in this picture. Right? So here's the cartoon version. What they notice when they look at the membrane-bound subunits. So let me just draw one of them. So my cartoon version of one of the subunits. So here's the membrane. And so here's the subunit in the membrane. This is one of the three. So this might be the purple subunit or the blue one or the yellow one, and the details over here. All right, so let's see. This is the P side. This is the N side. What you notice is that there's an opening. There's a cavity in the protein that's open to the matrix side of the membrane. And there's another opening here, cavity. All right, so an opening means that cavity means things can go in and out, and in particular, it's big enough that water molecules can go in and out, and protons can go in and out. Right, so these can go in, they can diffuse out. And it's the same thing over here. And protons are what really matter. Protons can go in, they can diffuse out. Okay? So there are these two openings. So there's an opening, and there's the other opening. Note that they're not connected. It's not a continuous channel, because there's no connection from this opening to this opening. So there's no tunnel or anything here. So there has to be, so the idea is that protons are going to come in through here, and somehow those protons have to get from here to here. Okay? What they notice is that there's a lysine. This is why I chose lysine for the previous questions. There's a lysine that sits at the base of this opening. That's that little blue thing. And it's at the base of this opening, so their hypothesis is that protons can flow in and they can be bound by that lysine residue. Okay? And right next to that lysine is a glutamate. Okay? That's the red thing. So the red is the glutamate and the blue is the lysine. So they're sitting right next to each other. And so that's going to make this lysine a stronger base, weaker acid, because when it binds a proton, it's going to hold on to it. Because the glutamate is going to help it hold the proton, right, as we saw in the previous questions. Okay? So this would be the structure of what we would call oxidized complex 1. Oxidized complex 1 is before electrons have been given to FMN. Right? So before it reacts with NADH, that's the oxidized form of complex 1. All right, so I think I've labeled all these features. There are the openings. There's the lysine in blue. And there's a lysine in this subunit. There's also a lysine in this subunit and one in this subunit. And each of those is next to a glutamate in red. Again, the fourth channel is different. And I think in this paper, at least they weren't sure. So let's ignore the fourth channel. We'll do 3 of the 4 protons and try to get an idea of how they get through. Okay, so far we've got a lysine that's going to hold the proton, and the glutamate's going to help it hold the proton, and this is going to be the oxidized protein. All right, the other thing that's shown in this picture is that there are some other lysine residues, and lysines or glutamates, that are here and here. So one of them is close to this opening, and the other is sort of halfway in between. Right? And they've drawn these as uncolored, implying that maybe they're not protonated, although I mean, presumably this one could be accepting and giving that protons, right? All right, so this is the oxidized form. Before electrons get in, this is the conformation, well, this is the conformation indeterminate experimental. And let's say that a proton has gotten all the way into that lysine. Now the question is, how is that proton, the idea is that a proton is going to get from there to there and go out. Okay? So I'll stop here, and I'll start with what happens when electrons from NADH get put into here and they travel down. Because that's going to initiate the conformation changes. And I've already kind of applied what happens. So I'll just summarize that quickly.
So I'm not going to be able to come on Friday. Can you take notes for me? Thank you.